Okay, there we go. Everyone can hopefully see that.
Yep.
Perfect. Okay, so thank you all for attending. This is a KBase webinar that was put together by chiefly Dylan Chivian. He made this narrative for you all to check out today. The title of it is Genome Extraction from Shotgun Metagenome Sequence Data. And it kind of gives you the overall guide to taking input metagenome raw read data and obtaining high quality genomes in the end. Here at the top, these are nice little cells here that you can use within the KBase narrative to indicate some comments and notes with your narrative. And so in this case, Dylan has constructed a nice little graphical representation of the timeline or of the app workflow that we're going to be using today. In the next cell, this is a bit of an overview. I'm just going to read explicitly. KBase has powerful tools for metabolic modeling and comparative phylogenomics of microbial genomes that can be used to develop mechanistic understanding of functional interactions between microbial species and their ecosystems. Central to this process is getting high quality genomes to annotate either via cultivation or genome extraction from metagenome assembly. KBase has a suite of tools meant to be used in concert. And here we're going to go through them. And after assembly and binning, high quality bins are annotated and they can be used in comparative phylogenomics analyses, like in other example narratives that are linked there, and through and for the purpose of metabolic reconstruction and community interaction modeling. And there's another narrative link there if you'd like to check it out. The two samples that are presented in this narrative today come from a compost enrichment metagenomes, two compost enrichment metagenomes, samples called 37A and 37B. And there's a link to the PubMed article if you all want to check out the actual publication related to these data. And there's a link to the Joint Bioenergy Institute, one of the Lawrence Berkeley Lab institutes. Moving on to the main lessons from the narrative, we're going to cover in a nutshell, I would say, like 8 things. We're going to learn how to perform quality control of read libraries. We're going to learn how to measure taxonomic population structure within environmental shotgun reads. We're going to learn how to assemble metagenomes. We're going to compare assembly quality. We're going to learn how to bin metagenomic scaffolds into putative lineages, so producing metagenome assembled genomes or MAGs. We're going to learn how to assess the quality of MAGs and extract those individual genomes. And then from there, we leap into annotating genes within those genomes and placing those MAGs into a reference species tree. Moving a little bit further down here, go to full screen. So here is a breakdown of the different narrative sections. The first section here is read hygiene. We'll focus on that first. And the other sections include classifying taxonomy, assembling reads, comparing the contigs, binning the contigs, assessing the quality of the bins, extracting individual assemblies, annotating genomes, and finding the relatives. And these are all hyperlinked. So if you want to jump rapidly to different elements in the narrative, you can do that. So I just click that and I'm going to scroll back up here. Just a word on timing. So KBase, it boasts faster processing and runtime over of many apps here over our competitors, but keep in mind that metagenomes sometimes can be rather large, and so they do take some time to analyze. The queue times for metagenome data can be very lengthy at certain periods, and once you have your data loaded and you're through the queue, we hope you enjoy faster runtimes of hours and days over the months it can take using other platforms to analyze these types of data. So this table below kind of shows a bit of a snapshot in terms of what one might expect with sort of standard metagenome input data. And this app Kaiju, which works on raw reads, it has a very short queue time and it runs about a couple hours. That's not a problem. We can run those apps in our sleep. The challenge becomes a little bit more when we have these applications that are doing metagenome assembly, which they can be in the queue for significant amounts of time. In this case, the average is almost 12 hours for metaspades. And they also run a fairly long amount of time. And so these are just, I guess, tips you want to keep in mind when you're trying to run a lot of your data. Of course, if you have many different sequencing libraries, you probably don't want to pick the tools that take the longest to run. So just some things to keep in mind as you're using the KBase platform. Here's a bit more thorough description of the apps we're going to be using in this tutorial. The first app is called Fastqc, which allows users to check the quality of raw sequence data from high throughput sequencing pipelines. And there's a little descriptor of what the output looks like there, but we'll get to that. Trimmomatic is an application that performs a variety of trimming tasks on paired or single-end Illumina reads. We'll use that to trim the bad quality regions of our sequence data. Kaiju is an application that generates fast and sensitive taxonomic classification of raw metagenomic reads by comparing those to a database of known microbial proteins. So we'll highlight that application and show how it can be used to infer the taxonomy in your unassembled data. From there, we'll get into 3 different assemblers: Metaspades, Megahit, and IDBA-UD, which all use various algorithms to assemble your metagenomic data into large contiguous pieces that we analyze downstream. We'll take the output of the assembly applications and we'll compare the assembled Contig distributions using a custom developed app that was built on system here. From there, we will use one of the Contig binners called MaxBin2, which uses nucleotide composition information, source strain abundance, and phylogenetic marker genes to perform binning using an expectation maximization algorithm. We have other binning tools on system, but that's the only one we'll be covering today. After we perform the binning, we'll assess the quality of those bins using the tool CheckM, which is a set of tools that can be used to assess the quality of draft genomes, such as MAGs. After we've assessed the quality and we determine what genome bins we want to keep, we will extract those bins and turn those into a useful object for downstream processing. From there, we'll have microbial assemblies that we can then annotate using tools such as RAST on system. Then after we've annotated the microbial assembly, we can kind of lump all of those genomes together into a set, is what they're called on KBase. And then from there, we will take this set and we will insert all of those genomes into a species tree in order to construct a phylogeny which gives a sense of the closely related species that you might have in your samples. And after the tree is generated, we'll view the tree using the app, and that allows you to look at and download standard formats like Newick for trees. Okay, so with that introduction, let's get into it. So the first step is read hygiene. So we begin by importing sets of paired end Fastqc reads for the metagenomes 37A and 37B. The import app, it creates these objects called paired end libraries. I'm gonna expand this box here. I clicked the upper right side of this box, and that gives us all the detail here. And so when we click the view configure box, you can see all of the parameters that were input here. It was indicated that's a Fastqc/FastA file. This was the name of the file as it was as it existed on the staging area that Dylan imported it from. Dylan indicated it was an Illumina sequencing read and then gave it a name in the end, and also indicated that it was interleaved. So he plugged in those configurations and ran it, and it imported the data successfully. We can take a look at some of the stats from the library that he imported here. We got 95 billion reads, give or take. It's a paired end library. And that's kind of the bulk of that information. And then some other info here to the tune of the total number of bases, the average read length, standard deviation of read length, the number of duplicate reads, the quality score average, GC percent, base percent, etc. Gives some nice information about your library. So that was the first sample, the first metagenome sample that Dylan imported. This is the second sample here, 37B. And I've gone over those details, so I'll just collapse that box. And you've seen an example from 37A of the stats that are produced. So we have the identical stats for this 37B sample. Okay, moving on to Fastqc, the first full KBase app we're running here. So this was run first on the 37A samples, and we can take a look at the results here. And Fastqc, the tool that provides you this nice report in the end where it gives you a lot of information about your read library, basic statistics that are not totally unrelated to the statistics that are auto-generated for you when you import your data in KBase. But some of the nice things that Fastqc provides that our KBase auto importer doesn't handle is it produces these nice maps of your read library quality in the form of highlighting positions in your reads that where the quality starts to flag. And so, you know, in short, this is a tool that allows you to sense where you want to quality control your data for downstream processing purposes. And so for those of you who are unfamiliar with interpreting these charts, the non-ominous red color here highlights that these are Illumina-based qualities that are not ideal. And therefore, you know, there's going to be some amount of trimming that we're going to want to perform likely on these reads here. And so there's various other information that you can extract from the Fastqc report. I will leave it open for you all to explore, but the most important information is getting a rough sense of the quality across the positions of your reads. Fastqc was run a second time here for the second library, the 37B library, and we have relatively similar information that we're looking at here. It highlights that there's some trimming that we could perform on the most distal end of these reads to perhaps improve the quality of them. Okay, so that takes us to read trimming. So read trimming is performed on system using a tool called Trimmomatic. Trimmomatic is a relatively flexible tool that allows you to manipulate your sequencing libraries in a variety of ways. Let's take a look at the parameters that Dylan used here. So he plugged in that it was a paired end library with a certain Illumina Fred type. And then he indicated that the sliding window size to look for, to identify frames for trimming is 4 here. And the sliding window minimum quality is going to be 15. And if you have questions about what are the details behind some of these relatively short phrases like sliding window size or sliding window quality, you can click on these terms here, and it gives you a nice pop-up box with a bit more information about the different parameters. So if you have questions about, you know, what are the different parameters being plugged into the applications here, you can do some investigating yourself to figure that out. And so it looks like Dylan trimmed with these 2 parameters plugged in, and it produced this library called 37A that's trimmed and has the name head crop 5 crop 140. And we're going to expand this other Trimmomatic box here, which highlights that essentially we did the same thing. Dylan did the same thing for the 37B sample here, which seems like he used the exact same parameters. And so, okay, that's great. Let's take a look at how that worked out. So after he ran the Trimmomatic tool, he reran the Fastqc tool that we've previously used to inspect the quality of our reads. And lo and behold, what the punchline here is that we've improved the quality of our raw reads by essentially trimming out the bad regions or the bad reads in their entirety. And so this is the error profile based on the Fastqc assessment, and it highlights that across all of the Illumina positions, we have overall better quality, and it seems like that's because we've trimmed at least the 15 base pairs off the end of the molecule here. And so there's an example of how using read QC tools gives us better reads to work with downstream. Here is the same result, I suspect, for the 37B sample, where the similar trimming parameters were used, and it probably gave us a better looking error profile. And yep, sure enough, we have more reads that are in the green region there, which is great. It's a much more realistic signal. Okay, so now that we have cleaned up our reads, performed read hygiene, where we can move on to using these reads in our downstream analysis. So one of the first tools that we can use here with the raw reads is Kaiju, and it predicts the microbial composition within your raw reads based on protein similarities. And so this is an advantage for people wanting to inspect the taxonomy of their samples without performing metagenome assembly, which sometimes doesn't always assemble every piece of your sequencing data. And so, okay, let's take a look at Kaiju. First, we'll take a look at the configuration here to see what Dylan used. Kaiju allows you to set multiple read library, input multiple read libraries at the same time, so you can you don't have to run many Kaiju apps in parallel here. So he inputted the two quality controlled 37A and 37B libraries. From there, the parameters he used, I think these are default parameters, but he's chosen to look into all of the taxonomic levels that are available. He used the Refseq Genomes database, which does not have any eukaryotes. And then these are default parameters, but they essentially do filtering on your data to minimize the calculations that are happening behind the scenes. That helps speed things up. Okay, let's take a look at the result. So Kaiju has a really nice graphical output. Dylan did a great job constructing this output, and it gives you a really good sense of the taxonomic classification of your samples here. Now, I'm not familiar with the details of these two samples or the study intricately enough to comment on whether these things are what we expect to see in these samples, but you do get a sense that there's a lot of diversity here, and that this is additional information one can use when trying to pursue their scientific questions. So there we go. The Kaiju output provides you taxonomy at all the different levels. This last one here is at the genus species level. So yep, that can be very useful. And if you want to download the Kaiju abundance and the chronoplots, there's chronoplots that are made under the hood here but not displayed. You can download that information, and you can download all of the data here as well. Okay, that is the only analysis we'll be performing on the raw reads. From there, we're going to be moving towards assembly. And so now that we have cleaned the reads using the Fastqc and Trim-O-Matic combination of tools above, we can assemble the reads into larger contiguous fragments called Contigs, and therefore that allows us to create an approximation of the scaffold of the whole genome, the metagenome in this case. KBase offers several metagenome assemblers and a tool for comparing their output, which is very similar to Quast. A couple notes here. Out of the different assemblers that we have, Metaspades only accepts a single library as input. So the app to merge multiple read libraries into one read library was used to combine the reads from these two samples and perform a co-assembly. So that's one nuance to keep in mind if you're using Metaspades. And another note, we ran the megahit assembler twice here using different parameters, but on the same input data. The first run was used to produce a using the meta large as a preset, which is a setting that's catered towards large and complex assemblies. And the second attempt that we have down here, it uses the meta sensitive preset, which gives a more sensitive assembly but runs slower. And so, you know, what we're trying to highlight here is that there's multiple ways that you can run your input data and then subsequently inspect your results. Okay, the first assembly that we have here, is generated by Metaspades, and this was the 37A library that was QC'd and trimmed. The default parameters plugged in here are 2,000 base pairs as the minimum Contig length, which is quite reasonable. And we have the Contig name, or the output Contig set name right there. You can click on the result box, get a quick sense of the results. So Metaspades, or all the assemblers, when they complete, they automatically run this QUAST tool, which gives you a nice thorough breakdown of your assembly statistics. And in this case, one of the things that there's many ways to go about assessing the quality of your library, of your assembled metagenome library, but oftentimes a metric that's used is counting the number of contigs that are largest in size within your metagenome. And from performing metagenome assembly, I can tell you that contigs above 100,000 base pairs are relatively large and somewhat rare in your metagenome assemblies. So having 162 of them is pretty good. We didn't get any sequences from this assembly over 1 million base pairs, but that's all right. We came close. We had one that was 940,000. Yeah, there's other statistics you can inspect here, such as the N50. This is the contig length, such that using longer equal length contigs produce half of the bases in that assembly. And these are all statistics that are typically used to describe the output of assemblies. We have a plot here showing the length of the contigs ordered by the rank, I believe this is here. So more information that you can use to inspect your assembly. Moving on to the next assembly here, we have the second sample, the 37B sample, and Dylan used the same parameters, named it differently, of course, and ran the assembly. And with this particular sample, there were, give or take, the same number of contigs above 100,000 base pairs, but there were, interestingly, 2 contigs here that were above 1 million base pairs. And so perhaps this is a pretty good assembly. Okay, the next attempt here with Metaspades, as we noted above, we invoke the merge multiple read libs into one read lib app. And this is required for running Metaspades with multiple read libraries because we don't allow multiple inputs there. So that's just sort of a unique caveat here, but this is how you would operate that on system. So Dylan has merged the 2 quality controlled libraries here into a merge 37AB library that named right here. And these are the libraries that he uses as input for Metaspades below. This is the output from the merge read libs to one read libs application. It just gives you a quick summary of that your read libraries were combined. And so now to what we were attempting here with the merging of the read libraries. Dylan ran the Metaspades app again, this time with the merged reads together and using the same parameter set and the appropriate naming scheme. And let's take a quick look at the result he has here. Okay, so in this case, there were almost 300 contigs that were above 100,000 base pairs, which is up from, what was it, like 180 to 200 or something like that from the individual assemblies. And we have 2 contigs in this case that were above 1 million base pairs. And so it's tough to say just looking at these statistics, but it seems like there are some pros to this combined assembly that Dylan put forward here. Moving forward through the additional assemblers that we have here on system, we have MegaHit, which let's see how Dylan ran this. He ran this on the merged output, the merge read libraries from the 2 samples. And this first attempt with MegaHit used the meta-large parameter for large/complex assemblies. We have a minimum contig length of 2000 and an appropriate output assembly name here. And we can take a quick look at the statistics of the result. So in this case, there were about 200 contigs over 100,000 base pairs and a single contig above 1 million. So, okay, that's our result. Now we run MegaHit again, but this time Dylan has chosen the meta-sensitive parameter preset, which results in a more sensitive assembly, but it takes longer to perform. He has the same minimum contig length and an appropriate output assembly name here. And here is the results. So interestingly, we have fewer contigs that were above 100,000 base pairs and 1 contig that was above 1 million base pairs, down from 2. And so interestingly, in this case, it seems that the sensitive preset produced contigs that were not as long. Okay, moving on to the third assembler that we're demonstrating here on system. This is IDBAUD. And in this case, Dylan ran the merged libraries, the 37AB libraries together with the minimum contig length 2000 again, and an appropriate output contig set name. And let's take a look at the statistics roughly. There are about 150 contigs above 100,000 base pairs, and there were 0 above 1 million. And so, okay, that's a third assembly. And so with all of these assemblies performed on system, it can be challenging to determine which set we want to move forward with. In this case, we have, Dylan highlights that we have 6 sets of contigs generated using various assemblers. And the goal here is to examine them side by side to determine the one that has the highest quality. Dylan has taken the statistics that are arguably highly relevant and summarized them here in this table here. The N50, which is the shortest sequence length containing 50% of the entire assembly, and the L50, which is the least number of contigs whose sum length is equal to N50. And so taking a look at the output here, we can evaluate the statistics, and Dylan has come to the conclusion that the assembly from running metaspades on the combined library generated the highest quality contigs, and therefore it will be used for further analysis. And so the highest quality here is probably rooted in the sum of, in multiple things, but I would say probably rooted mainly in the sum of the length of the contigs that were produced. So it just means that metaspades assembled the most number of pieces successfully together. It also had the most number of contigs above 1 million base pairs. So all good things for metaspades. And indeed, it is a popular tool because it does seem to work really well. Okay, that takes us to this next app, which is a very handy app to look at the statistics and assembly qualities across your different contig sets. This app is called Compare Assembled Contig Distributions. You're already getting a sense of the result there, but let me click on the configuration so we can see what Dylan put in here. So we put in the 6 different sets of contigs, excuse me, the 37A, 37B, and the merge 37AB all run with Metaspades. He has the 37AB merge reads with the 2 different megahit presets, and the 37AB with the IDBA UD tool. All right, pulling up the results here. And so there's a lot of interesting graphics that pop out of this app. These visuals are useful in trying to understand the usefulness of your assemblies and to get a sense of how your assemblies compare to each other. And overall, what you're trying to look for is to have the sum of the Contig lengths be highest in the Contigs that are ordered from longest to shortest. And so, in other words, the more upward trajectory is desired in looking at these plots here. And in this case, the 37AB merge library run with the Metaspades assemblers produced the best assembly, and you can see that here on the graph. There's many other cool visuals here that Dylan has placed into this app to highlight your assemblies and your statistics. So please check those out. It's a lot of great information that you can use to determine what data products of yours you want to move forward with downstream. You can download all of these plots and all this information in PDF and PNG format as well. Okay, from there, we will move into binning contigs, which is an exciting thing that people do with DNA metagenome assemblies nowadays. And at this point, I think I am supposed to pass this off for Dylan to move forward with the narrative from here.
Actually, Sean, why don't you take it through up to the phylogenetic placement? Is that cool?
Yep, you got it. Okay, so let's talk about binning contigs. So once we have our metagenome assembly created, we can we can sort the contigs that have been assembled into rough approximations of the individual microbial genomes that were in the sample to begin with. And there's metagenome binning is a major topic right now. It's under active development in the research community, so I can't do it full justice here, but it works really, really well. And there's been a lot of recent scientific publications that have used genome binning to reveal new ecological roles in the microbes in the environment and in other types of samples. So check it out and use it. It's a fantastic tool, a fantastic procedure to employ on your data. The tool that we're using here in this tutorial for Contig binning is called MaxBin2. This one was developed at JBay, I believe. And we're using the, let's take a look at the configuration here. We're using the merged read library assembly from Metaspades here as input. And then we are also using the individual read libraries as input, the founding libraries that we use to develop this co-assembly as input. And then an appropriately named bin contig object name here at the bottom. And we run the app, Dylan ran the app, and this is the result. So out of binning our metagenome assembly, we obtained 65 genome bins from a starting set of 27,500 contigs. Majority of those contigs were binned, about 85% of them, which is fantastic. And only about 14% of those contigs were unbinned. The number of contigs that were too short were 0, which is fine. We have no problem there. And then it gives you a couple other statistics here, such as the sum length of the bin contigs, sum length of the unbinned contigs, etc. But overall, this is a pretty great result. We have 65 putative microbial genomes to interrogate downstream. And you can download the results of the MaxBin app and this PDF output of the marker genes that's produced by MaxBin also. Here is some additional information that's produced when you run the MaxBin app. It gives you, again here, the number of bins, the total number of nucleotides represented in those bins. And then if you click this bins tab here, you get a rough sense of the breakdown of marker gene completeness from running MaxBin. By bin, you get a sense of the GC content, and the number of contigs, and the total contig length in each of these genomes. And this can be useful information to help you QC your data. You know, for example, the total contig length here should approximate something close to what a microbial genome size is supposed to be. The number of contigs here, ideally, you have a lower number. That means that you have, you know, less chances for having contigs be binned incorrectly, etc. So lots of good information for you to explore. Okay, so how do we assess the quality of bins? Well, quality control is a necessary step at every level in this analysis, and I would say, you know, especially here with respect to the quality of bins, because, you know, we want to make sure that our genomes are what we say they are before we perform a bunch of downstream analyses. And so let me read this note here. So from the graphical output below, we see that there's 64 total bins. There's that I'm given kind of the punchline here of this next app we're going to run. 28 of them are above 90% complete and less than 2.5% contaminated, which means that they're very high quality genomes. And these are the numbers of the bins that correspond to those statistics. And so how do we generate that information? Well, you ran this tool called CheckM on system, which is relatively straightforward in terms of having a very limited set of input parameters here. You just input your bin contigs object, which is the output object that was produced from the MaxBin2 app up here. And Dylan just ran the default parameters here, which gives you a reduced tree and saves the plots. And let's take a quick look at the result. So for those of you not familiar with CheckM, CheckM is a powerful tool developed by the folks in Queensland. Phil Huggenholtz and Gene Tyson, Donovan Parks specifically is the lead designer. And it's a great tool that's used to assess the location on the tree that your genome is placed, and provides contextual single copy marker genes to determine if your genome is complete or contaminated. And it works fantastically, and it's very widely cited. So this plot gives you a rough sense of genomes of the marker genes that were discovered inside of your different bins here. The graphical input is custom made by Dylan. Graphical output here. Here's a key. So the green indicates these are single copy genes. The gray indicates those were missing genes. The hues of blue indicate the heterogeny, the strain heterogeny within your genome, which is a measure of potential strain level contamination in your genome. We can talk more about that if there's interest. And then also in sort of the red/yellow hues, this is the contamination portion for your genomes. These are contigs that are mixed in with your bins that should not be there, essentially, is the inference. And so CheckM produces this table of output, which highlights the marker gene lineage that was used to estimate the completeness and contamination scores, the number of genomes that were used in that inference, the number of markers used in that inference, the number of, I think that this is I guess I'm not clear on how marker sets and markers are different. I'll have to get back to that. These numbers highlight the numbers of single copy marker genes that were identified in the bins. And then at the end of the day, this is probably, as far as most users need to look, the completeness and contamination numbers that are calculated from all of this information are presented here. And so completeness numbers that are very high, approaching 100% are ideal, and contamination numbers that are very low, approaching 0% are ideal. So this would be a pretty darn good genome bin right here, mostly complete and free of contamination. This one right here, it's a little bit tougher. It's only 76% complete and has a bit more contamination. So, if you're investigating this bin, these are just things that you need to keep in mind. And so we have this information for all of these different bins, which is fantastic. Scroll to the bottom here. And you can download all of this information and the outputs from the Check'em tool here. Okay, once we have identified the genome bins that are of high quality from running Check'em, we identified 28 total that were of high quality in the above. And we are going to be extracting those now as assembly objects. And this is achieved by running this tool called extract bins as assemblies from bin contigs. And so this will take a bin contig object as input. This is the object that was produced by MaxBin. And we have indicated that we are going to, Dylan indicated that he's going to extract, in this case, it seems like all, or 28 bins here. And so that's great. We've, from the starting set of 65, we've selected our 28. And then we click run on that tool, and that produces the assembly set that we want to use on system. Okay, so that takes us now to annotate genomes. And so at this point, we have high quality bins that have been extracted, 28 in total. And these bins now exist as assembly objects on system. Specifically, the assembly objects are all lumped together currently in the form of an assembly set, which is great because it allows us to operate on all 28 assemblies at once. And using this assembly set as input, we'll run the Annotate Multiple Microbial Assemblies app to annotate them all using the Rapid Annotation Subsystem Technology RAST pipeline. And a couple notes here: if you just wish to do a limited number of annotations, you can run them separately with the Annotate Microbial Assembly app. So if you just want to do, like one genome at a time, one assembly at a time, you can do that as well. One note here: once the high quality bins have all been annotated, those are all combined into a single genome set, and then the genome set is what we'll be using downstream for the remaining applications. And yeah, this tripped me up also. Just to keep in mind here that the genome set is labeled bin 001 through 65, but it only consists of 28 of the high quality bins. So just, a point for good documentation and keeping good notes. So running the Annotate Multiple Microbial Assemblies app, Dylan inputted the extracted bins.assemblies set object that was created from this extract bins as assemblies from bin Contigs app. And I suspect he probably just used default parameters within this advanced drop down here, and he named them appropriately this bins 1 through 65 genome set. So here is the result. It's going to be a lot of information. There's many different tabs to explore, many different pages, and you can go into various levels of depth. Here's a summary, mostly high-level summary, but it does go into, I guess, details on a per bin type of basis. Gives you a sense of the number of coding genes, number of CRISPR arrays, spacers, repeats, prophages, number of RNAs, etc. lots of good information that one is going to be interested in using when they're performing genome annotation. And from there, now that we have our genome set generated, we essentially have, just to recap, we've taken our raw reads, we've assembled our metagenomes from those raw reads, we've binned contigs from the metagenome assembly that resulted, we quality controlled the genome, the genome bin objects, we extracted them to prep them for annotation via RAST. And now we have a set of 28 high quality bins that have all been annotated with RAST. And with that, we're ready to run the next set of apps, which include finding relatives and inserting into the species tree. And I think I'm supposed to hand it over to Dylan at this point.
Hi, can you hear me?
Yep.
Okay, good. Sorry, I'm coming back from the questions and answers document, so my head's a little bit there. Switching gears, I've been watching Avenue 5 lately, and there's a character on it who, he's British, but he's convinced everyone he's American, and he has to remember who he's talking to figure out which accent to use. And I'm always worried with these things that I'm going to accidentally, I don't know, have some mental breakdown and start using an accent that doesn't belong to me and then be stuck with it. Anyway, hopefully this is the right one. Okay, so this is, where are we? Insert set of genomes into species tree. So once we have these mags, we want to make sure that, actually, can I do screen share here? Let me do this. All right, okay, no more answering questions. I'm cut off. There you are. Okay, so where were we? We were down phylogeny, right? Nope, that's too far.
Tree. Yep.
Okay, we've annotated, and now we're finding relatives. Okay, let's do this. Let me shrink that. By the way, I have a new background for these meetings. It turns out that video conferencing was predicted in 1940. So this picture is from a popular mechanics prediction from 1940 of video conferencing. So anyway, we're going to find relatives for our the MAGs, the metagenome assembled genomes that have come out of this. And we're going to start by putting them all into the species tree against the refseq genomes. And so that's what we'll do here with this insert set of genomes into species tree. And we're going to take the genome set from that annotation, bins 1 through 65. We're going to take all of them. And then because we have 28 members in there, we're going to set the neighbor public genome count to 28. It turns out that doesn't quite work as well as we'd like, and I'll explain why. And we're going to get out an output tree and a genome set that corresponds to that species tree. So if we look at that result, we we have something that looks like this, where we have our query genomes, the bins, colored in yellow here, and then the neighbor refseq genomes, the proximal refseqs, not colored. And so we can see that some of them are getting a neighbor, and some of them are getting lots of neighbors, like this Rhodopseudomonas for bin 19. But then others are, not getting so many. And there's a reason for this, and that is because of the algorithm is greedy to distance. And so when we have members of this that have lots of species members that are from RefSeq that are close, those wind up sort of consuming the neighbor count. And so we need to sort of enforce some more diversity. And we're going to do that kind of manually. Additionally, we can see, and there is value in doing this, we get some of the proximal RefSeqs, but we also can see how our bins are grouping in the species tree as well. So this, we can identify this as a clade that we can then follow on the analysis against. So we're going to split them into clades. And so here we indicate which ones using the species tree. Clade A, we're going to call bins 26, 56, 5, and 35. Clade B, Clade C, Clade D, Clade E. Okay. So that we can accomplish by just doing this build genome set tool where we specify which genomes we want in that clade, maybe add a description, and then name it, clade A.genomeset. And we're going to do the same things for B, C, D, and E. So I will skip those. And now when we go and insert clade A into the RefSeq species tree, we get a much clearer picture for clade A. It splits things up nicely, and we really get a lot more proximal RefSeqs in there. And so we're going to do the same things for B, C, D, and E. And now we've got nicer species trees and more proximal RefSeqs. However, that just tells us our proximal RefSeqs and gives us sort of a local region of the species tree. If we want sort of a more comprehensive picture of where these bins are falling into the species tree, we can use this other tool where we are going to add in, it's basically a computational spike in of phylum exemplars to provide a skeleton. So the way we're going to do this, we're going to take our full genome set this time, not split up into clades. And we could have, if we'd saved the RefSeq proximals, added those in as additional genomes here. And so we could have both of them in the tree. And we're going to go and pick bacteria, archaea, and isolate, both isolates, mags, and sags as the phylum exemplars, and build a tree that includes both of them. And so that winds up looking like this. Probably should have stayed on that tab. There we go. So if we view it in a separate window and zoom in a bit, we can get a better picture of it. So here in violet, in this case, we're coloring the phylum exemplars. This is the species skeleton or phylum skeleton for the tree. And now we can see where our bins are falling into phylogenetic space more clearly. And then we can see that a lot of these guys are grouping together, but now these are, they're actinos, good. And so on. Bacteroides chlorobi, we're having quite a few of those, and so on. So there's sort of this balance we want to achieve of the sort of high-level global view of phylogenetic placement, and then the more high-resolution, zoomed-in view that you get from the truly proximal RefSeqs, right? And then you might take these forward with doing comparative analysis on the proximal RefSeq against your bin for functional completeness, you know, anything that looks like it's part of the greater family as a core functionality, or what's special or different in terms of presence in your bin functionally. Absence can be a bit trickier because, you know, these things are usually not circular genomes, although that's starting to be the case more. And so you wouldn't necessarily trust absence of functionality until you have a circular genome. But anyway, that's a whole nother topic in phylogenomics and functional assignment, and we will do webinars on that later in the summer. But for right now, that's beyond the scope of this one. So, so where did we get to? Well, we took shotgun metagenomics from 2 samples, and we were able to identify the lineages that were present and abundant in them. We were able to assemble those, and we were able to extract the mags from those and do a phylogenetic placement of them. And so those mags are really sort of the best part of the microbiome here. And so we really do want to put most of our effort initially into getting really high quality genomes there, because that's going to give us the best opportunity to analyze them as though they were isolate genomes. I mean, in essence, bacteriology started obviously not with environmental shotgun sequencing, but rather, taking a sample and streaking it onto a plate and getting colonies, and whichever grew best, that's what you targeted, right? And so, and that still goes on today. And it's an effective technique because it tells you who grows well, and you can really, maintain a clonal population of that. But a lot of times what you're obtaining from your wild sample is just those lineages that are sort of self-starters, fast growers. And so the reason we have turned to environmental genomics is a lot of organisms have interdependencies, and you can't just isolate them so easily, at least not without knowing what their metabolic dependencies are. So this approach to getting mags is really very useful for finding the more abundant guys that won't grow on their own, right? And so there's an assumption that if it's abundant in the wild, that therefore it's functionally important, and that may be the case. It's certainly contributing more to the metabolic capacity and overall community phenotype in a lot of ways. But these are the mags that we get out. However, it's only a partial picture of the microbiome. Sequencing is only a partial picture of the microbiome, right? I mean, we are not sequencing comprehensively every cell to the point where we get the full genome out for it. We get as much sequence as we get, and then anything that we didn't sequence, we don't see, but it's still there, right? So the best we can hope for is being able to analyze all of the sequence and each fraction of that sequence the best we can. So high quality mags, the best fraction, deserve the most attention, but they're only part of the picture. So I want to talk to you a bit about where we're trying to go. So this is early work. It's not yet fully available, and a lot of the downstream stuff is not available yet, but it is coming online soon. And so we'll have a webinar on those details once it becomes available to everyone. So this is the picture. And again, this is just for the sequenced portion of a microbiome. This may represent, less than 10% of the actual wild community, but it's what we can get. So if you take the entire set of sequenced information and, draw that as an ellipse and do a Venn diagram that wound up looking a bit like an alien. I meant it to be more like a Matryoshka doll, but it wound up looking like some sort of weird alien to me. Anyway. And so we take that and we do it as a sort of nested Venn diagram. We have a fraction here that's the unassembled reads. Actually, let's go the other direction. So we have in the very sort of most valuable fraction here, the high quality bins. And so we're going to really want to isolate those out. And that's what that entire previous narrative was focused on. And furthermore, the fraction in high quality bins, actually, these are being further fractionated into individual bins, right? So you can actually think of a bin, yes, it corresponds to a lineage, but it's a fractionation of your community into one lineage, right? And so we're getting the bins themselves wind up being a fractionation of the entire community. And then there's this sort of greater set, which is the high quality bins together. But after that, there's still value in the medium quality bins. So say they're 50% complete and less than 5% contaminated or something like that, meaning the assembly and binning process may have produced a small amount of chimeric contigs, or some of the contigs may have been thrown into the bin by the binners, but actually belonged in a different lineage. That's the concept of contamination in a bin. So these are pretty good, and it's sort of wasteful to just chuck them out and not take advantage of the sort of co-occurrence of genes that we get from the medium quality bins. But we have to be very careful and certainly make no statements about absence of function statements. And then there are the low quality bins where it's sort of below, arbitrarily, it's up to you what you decide is a low quality bin, but, to just throw a hard cutoff there, it would be, say, less than 50% completeness and greater than 5% contamination, something like that. And really at that point, you trust them so little that you might just throw them back in with the unbinned contigs and just say that these two represent a fraction together that you want to analyze. And that would probably usually be the case. However, if you really want to work at it, you might take all the low quality bins or the low quality plus the medium quality and start to see whether or not some of those could be coalesced and make a high quality bin. So for example, you may look at the lineage assignment, and you could do a pan-genome analysis of all isolate genomes for that clade and discover what the core functional markers are for that lineage. And that's actually how Check'em essentially works, right? It's developing lineage-specific, clade-specific markers based on pan-genome analysis. So you could do that for your medium and low-quality bins, and then look at which ones are complementary to one another, and start merging them. But it's very labor intensive and requires a lot of expert information and attention. So if you were just going to go forward with automated analysis, those low quality bins should probably be grouped with the unbinned contigs. So, all right, so what do we wind up with for sort of fractions for trying to analyze going forward for taxonomy and function? We have our high quality bins, and we're going to use our standard isolate genome analysis tools. We can do metabolic modeling, we can do pan-genomes, we can do comparative phylogenomics with those, always with the caveat that, yes, we know these are MAGs. Medium quality bins, we can do something somewhat similar analyses. We may want to take them and do the complementary merging, things like that. We definitely want to treat them as a separate fraction. And then the low quality bins, unless we're going to do some manual stuff, we're going to treat those as the same as unbinned contigs. We're going to throw them right back. Unassembled reads, there's value there. You have to be very, very, very careful. A lot of the reads represent fractions of proteins, and so the assignment of them is less trustworthy. Anyway, right now we don't offer a tremendous amount of functionality for unassembled reads other than the taxonomic placement like Hygiene and that sort of thing. But again, remember, they didn't assemble, which means they didn't have a lot of read depth in that lineage, which means you're not going to get multiple observations that support an assignment of a lineage. So it's probably going to wind up a bit scattered, a lot of the tail, that sort of thing. But it may be numerically abundant collectively, right? It may carry important function collectively. So you can't just discard it. We need to pay attention to it and appreciate it. Okay, so I'm not going to get into the downstream functionality in terms of taxonomy and functional assignment for each of these fractions. That'll be a later webinar, probably later this summer. Stay tuned. But I will tell you a little bit about fractionation tooling itself. So we have a lot of utilities for handling sets and different data types. We saw in the previous narrative, merging reads libraries, stuff like that. It's, merging, fractionating. These are utilities meant to simplify that process. So one of those tools is we're going to try and split off just the high quality bins. So instead of taking all of the bins, we're going to split off the high quality as a separate fraction. And so for that, we have this, we're going to use Check'em because we want the completeness and contamination scores from Check'em to do our fractionation. And so this essentially runs Check'em, but with an extra couple arguments of completeness threshold and contamination threshold. People have different values here. I wanted to be really strict, and so I went with a 2% contamination. That's pretty low, and the fact is a lot of the definition of completeness and contamination is based on pangenome analyses of isolate genomes sequenced to date. That's only a portion of what's out there. And so our picture is improving rapidly, but there's still less well-sampled regions of the species tree for isolate genomes in particular, and therefore our ability to get meaningful pangenome analysis is limited. And so the definition of a clade-specific single copy marker or is, because it's dependent upon that, maybe an incomplete picture. And so doing hard thresholds like 0% contamination is not to be trusted. There are additionally lineages that should have what are supposedly universal housekeeping genes and therefore universal phylogenetic markers, and they don't have them. Sean can tell you all about that. He's really been looking at that quite a bit. And so you can't just say, oh, it must be 100% complete. It must be 0% contamination. Unless it's, E. coli, maybe. But even there, I wouldn't trust it. So we're going to get out a bin Contigs, but it's specifically going to be high quality bins because we're imposing these thresholds. We're using the same input set. In this case, we're taking in the bin Contigs that had everything from MaxBin, and then we're going to filter with the Check'em. Let's see, I've forgotten what my advanced parameters. Reference tree, so same as Check'em, and save plots. Yes, okay, that's the same as regular Check'em. And the result is also pretty much the same as regular Check'em, except we get this binned Contigs object out of the high quality bins. Okay. Next, we're going to take those, and now since this binned Contigs object is specific to the high quality bins, now when we do the extraction, we just extract all of them. That's the default. We don't have to manually pick which ones we want because we've already filtered to the high quality bins in this bin Contig. So the genome set that comes out is going to be the high quality extracted. Okay. We get individual elements of the assembly set and an overall assembly set that contains the links to these. So now this assembly set is what we're going to want to use for batch processing downstream. So, we follow these steps in the previous narrative, but just to reiterate them here, we're going to take that assembly set as the input for genome annotation with Fastqc, and this is multiple microbial assemblies, so it's a batch annotation. We could have individually specified the genomes in the assembly list, but since we have the assembly set, we're just going to use that for configuration. There's no scientific name since it's a collection of genomes, and we're going to get a genome set out. Ordinarily at this point, I would take that genome set, I would put it into a species tree as we saw before, in the previous narrative. I would go into comparative analysis, functional analysis, using the phylogenomics tools in KBase, but I'm not going to do that today. Instead, let's talk about getting the medium quality bins out. So here we're going to, I realized I should have done the parameterization a little bit differently when I went to do this. It's one of those things, whenever you do something the first time, you think you've got the complete picture, and then you go to use it a slightly different way, and you realize you needed to change something. So for right now, there's just a lower bound on the parameters. So completeness we're setting to 50%, contamination 5% now. But that's going to give us everything above that. So that's actually going to wind up including the high quality bins. And if I just included an upper bound here, then we would get just the medium quality slice that we were targeting. So I'm going to add that. Pretend that we have it today, because we'll have it tomorrow. So lower bound completeness 50%, upper bound contamination 5%, and then we're going to wind up getting this whole fraction. So it'll be medium quality plus high quality that comes out. And now we're going to go and extract those into assemblies. And again, it's going to be medium quality plus high quality. We're going to batch through those. And we're going to wind up with a genome set that's the medium quality plus the high quality. Now we want to split off the unbinned contigs and the low quality bins, because we've decided we're going to group those together from the medium and high quality bins. So we can do that with this Fractionate Contigs tool. It's a utility. And so we can put in our complete set of contigs. Here we're using the 37AB Metaspades contigs that we've been using, for everything. That's the full set of contigs, not the full set of metagenomic data. They're still the unassembled reads, but it's at least all of the assembled information. And now we're going to use as our positive filter the medium quality plus high quality assembly set. This could be a binned contigs object. It could be a genome set, as long as the contig IDs are the same between the initial assembly and the downstream data product. That is true in KBase. If you do your processing in KBase from contigs through to genomes, we keep the same contig IDs through the bin contigs, through the assemblies, through the genomes. So you can use those as your positive filter objects. In this case, I'm using the assembly set. It was kind of arbitrary. And we can choose whether or not we're going to just pull the negative. So this is a Venn diagram, but it's a fully contained Venn diagram. So we can take either the negative fraction, well, I guess it's the orange part here, the negative fraction, the positive fraction, or both. And so in this case, we're going to split it into both for a reason that will become clear below. And so we're going to output this 37AB Metaspades versus the positive fraction contigs, and it will append negative fraction or positive fraction to the object name automatically. We get some information too in the results. The created objects, positive fraction, negative fraction. So this corresponds to medium and high quality. This corresponds to low quality and unbinned. And then some basic statistics. How many contigs are in the full set and the sequence length of those contigs. The positive fraction, number of contigs, sequence length of that, and so on. So this is actually a little bit interesting, and it conforms with what we expect. There are fewer fractions in the medium and high quality than the unbinned plus low quality, about half. But the sequence length found in the medium quality and high quality is higher than the unbinned. So that's telling us that there's more short contigs in the unbinned and low quality, and we're doing a better job when it's the longer contig of binning it. This conforms with expectations. We expect more phylogenetic markers and longer contigs, and therefore better binning. Okay, well, now we're at least going to split the medium quality and high quality bins. So here in purple is the medium quality, and blue is the high quality fractions, and we go and we're going to, as we saw above, we saved the positive fraction of medium quality and high quality contigs. So because we saved that, now that becomes our input for splitting. And this time we're going to use as our positive filter just the high quality. In this case, like I said, it could have been the bin contigs object. It could have been the genome set. In this case, I picked the assembly set arbitrarily. So now we're going to do a both split. And so we're going to get the positive fraction will correspond to the high quality, and the negative fraction will correspond to the medium quality. So it's, this iterative fractionation. We have to pay attention to which chunks are in that we're dealing with in each of these input objects. That's sort of why I've made these figures to remind myself of which I'm using. And then, so that split, let's look at the statistics here. And okay, so we had the 9,727 contigs with all of the sequence. And now the positive fraction, the high quality, fewer contigs, but about the same amount of sequence. So longer contigs. High quality yields, longer contigs yield higher quality mags. This seems to be the case. Kind of makes sense. All right, so now we have 3 fractions, and we're going to take a look at the contig length distributions in them. We sort of got a rough picture of it from these statistics in the output of the fractionation operation, but now we can take a look at their distributions, and we've got this compare assembled contig distributions, which is the Quast-like app. So these top two figures are very Quast-like. And we're putting in the complete set of 37 AB in blue here. So yes, we look at how much we're getting from the full set, and yes, of course, it's much longer. So now these are the fractions of it below, and we see that the positive fraction of medium quality versus high quality, in other words, the high quality fraction, in orange here, doesn't have the most sequence, but it is the longest. After that is medium quality in green, and then the low quality mags plus, low quality bins plus unbinned contigs in red. is the longest. Okay, so, and then we have the cost like N50, L50, 75, 90 stuff here, longest Contig, and so on. But the part I'm interested in looking at is the histograms here. So here's the full set, and we've split it up into Contigs of length under 10 kilobase pairs. When we did the assembly, we arbitrarily set a cutoff minimum of 2,000 base pairs so that we wouldn't spend all of our effort and all of the subsequent analysis dealing with very, very short contigs. Those tend to be the bulk of most assemblies. And unless you're looking for, small RNAs or transcription factor binding sites or CRISPRs, shorter contigs are typically less valuable for protein analysis. Protein domains are typically 100 to 400 amino acids. So typically at least 300 to 1,200 bases, and that's per protein domain. Many proteins are comprised of multiple domains, and so get much bigger. A rough estimate to a standard protein coding gene size would be on the order of 1,000 bases. So we picked 2,000 bases as sort of an arbitrary default that can be reconfigured to try not cutting the protein on average. But you can play with that parameter if you want longer ones. I do recommend using it though, because otherwise your assembly may contain quite a few, 500 base contigs, and it just chews up all your analysis. Okay, so the bulk of the distribution is in the short ones, as we said, even when you crop it at 2,000 bases, over 6,000 contigs, 1,500 between 10 and 100, and then a much smaller number above 100,000 bases. So now if we look at the high quality, so this next chunk is the high quality fraction. Almost none of that is from short contigs. There's more coming from, here, let's just compare them side by side. So this is the full distribution. This is the high quality fraction. Some of it, is coming from that medium chunk of contigs, and then, but a very significant chunk of the longer contigs are winding up in the high quality bins. So that means the binning is working well, and that's very good news. Now let's move on to the medium quality fraction. We're getting some more of the short ones, definitely some more of the medium ones, less of the very long contigs. So that's good. And this really does point to the fact that we didn't really want to throw out those medium quality mags. There really is some good stuff in there in terms of genomic context. One of the questions that was in the document was, why do this at all? Basically was the gist of it. And it's a great question. The reason is contigs that are longer give you full-length genes. They give you genomic context, so you can, if it's bacterial or archaeal, you can get operon structure, polycistronic elements, or genes that are typically conserved in their neighborhood. Often indicate system components of the same pathways, or if they have the same promoter region, that's a strong clue. Anyway, genomic context matters. Synteny matters. It's very useful and as a signal. And then the longer your contig, the more reliable your phylogenetic placement is. If you're doing a gene-based phylogeny following a Megan-like approach or something like that. And especially if you get phylogenetic markers in there, like a Check'em-like approach, that's very valuable too. So anyway, there's a reason to do this. And at the end, like I said, if you have a high quality mag, you can start to really, with caveats, pretend it's an isolate genome and follow isolate genome type analyses. Okay, and then let's look at the unbinned and low quality. As expected, most of the contigs in that portion are of the shorter variety, but we actually still are getting some medium length, 10 to 100, which is pretty big. Well, maybe not so many 100. My eyesight's not good enough to tell if there's anything in that tail, but we're getting some longer ones. But here's the amazing thing. Some of the unbinned and low quality bins are in fact these longer contigs, and they, I guess, did not comprise a, probably many of these were the low quality. I'd have to go back and check. Chances are some of them had some phylogenetic markers in them, but not enough to exceed that 50% threshold, and so we're dumped. Here's an amazing thing. This 1 point, I don't know if you can see this, but there's this 1.4 megabase contig that was not did not make it into the medium and high quality. So that deserves taking a look at. It may be a chimera. It's possible that it exceeded the contamination threshold. I would want to pull that one out and run that through Check'em by itself and see what happened there. So it may be a contamination issue, or it may be just it wound up being a standalone. But the fact is, because it got so long, the percentage of the community is probably great enough that it led to a great enough read depth for a long assembly, and you would expect there to be other contigs that also arose, and so there should be more of that lineage genome that came out. So my guess is contamination, and it's a chimera. That's my guess. Okay, so now we've split up the contigs. And like I said, you can go and start using some of the metabolic modeling, phylogenomics, functional assignment on the MAGs. But what about the other unbinned and low quality contigs? We know there's value there in genomic context and full-length genes, and now we can maybe use them for trying to do some mining for different functions. We want to know, which proportion of the community is contributing particular functions of interest. Obviously, we hope that it's mostly in the MAGs, but it may not be. It may be in some of the rest of the soup. So being able to do functional annotation of those contigs and start to get some understanding of what's happening in the soup is definitely important. There's a lot more tooling that we're working on today that I'm not going to share with you, but it will be coming online soon, and hopefully it would be later in the summer that we can share that with you. For right now, let me just say that what we can do today is we can take those contigs. So this is the low quality bin and unbinned contigs, this, the orange fraction, and you can build what's called an annotated metagenome assembly object. So we could have run this on the whole set right after we got the metagenome assembly. But then we would have wasted our high quality and medium quality mag fractionation. So it's better to do the binning first. And follow this approach that I outlined above, where you split off the higher quality and medium quality mags as really just more valuable data fractions. And then what's left, do the metagenome annotation to get these contigs, annotated contigs, annotated. And so then you can deal with this fraction and obtain the value from it, which is significant, but it's, like I keep saying, the high quality mags are, you can get more out of them. So, but we still want to get stuff from this. And so we're going to go and we're going to, in this case, use Proca to annotate these guys. This is a beta, by the way. So any of the annotated metagenome assembly stuff, like all our beta tooling, bear in mind that it's not fully vetted. We do encourage you to use it and use our help desk ticketing system to provide feedback to us, and we would really appreciate that. But bear in mind that it's just not done yet. So here we have the unbinned plus low quality fraction from 37AB co-assembly. And it's an annotated metagenome assembly object. And so we can move forward with doing functional analysis and even taxonomic analysis on those guys. So that will be a topic for another day. And I will stop sharing and maybe, I don't know, is there, Ben or Sean, is there anything else you want to say? Otherwise, we can maybe jump into the questions for the remaining 23 minutes.
Yeah, that's what I was just going to say.
What do you guys want to do?
If you wanted to maybe look over some unanswered questions and or, you know, offer some more depth on some of the interesting ones. Sean, I don't know if you had other ideas, but
no other ideas. Questions sound great.
Okay, so let me, all right, I'll reshare and pull it up. If I can find it. Where are you? That's it. Okay. I don't know. Most of the questions at the beginning are probably answered, so most of the questions at the bottom are probably not. Oh, definitely not those. So I'll work backwards since these haven't been answered yet. Why would you use Proca to re-annotate low quality and unbinned contigs rather than RAST? You could use either. I found like I said, this stuff is beta, and we're working on the RAST version right now. And last night I found a bug in it, so I didn't use it. I think that Proca is currently just slightly more bulletproof, but we'll get them both working well and release them together. So it's a personal preference. The reference data is different, the algorithm's different. You might do both and then compare the results. Depends on how much patience you have. I should say that the more you run heavy applications, like assembly in particular, the lower your priority, so your wait time in the queue will get longer. So it's a bit of a catch-22. I do recommend comparing things, but bear in mind that the more you run, the lower your priority. So do it judiciously if you're going to do it. Can microbiome fractionation narrative also be linked above? I'm gonna hide it again until it's a little more mature. It's really very developmental, and I wanted to share the thoughts with you. But I would prefer to it to be a bit more mature before making it public again. Once it's truly public and released, it certainly will be available and linked. But this was sort of a sneak peek. Is the CDS annotation of unbinned contigs trustworthy and can still be used? So CDSs are coding sequences, so protein encoding genes. Nothing's trustworthy 100%. So it works pretty well. Gene boundaries in a long enough contig, you get some gene chopping at contig edges, but hopefully those contigs broke because they're at long repeats like transposons or well, hopefully it broke on a transposon and not on a ribosomal operon, but it could have been. Anyway, but protein coding genes typically, hopefully won't be broken at the contig edge. And so because of that, the gene callers are probably going to work pretty well. And once you have a full-length gene, it really comes down to, it's no different from an isolate at that point for having functional or the classification of that protein coding gene. That said, isolate functional annotation is, what is it? E. coli is maybe 50% of the genes are well understood. And then I don't know what the numbers are today. But anyway, once you start getting into novel lineages, nothing is trustworthy. So yeah, Morgan Price, one of my favorite quotes from him was, there are lies, damn lies, and then there are gene annotations. So that's the answer. Question, is there something similar to the step of grouping bacteria or archaea based on 16S, but with another functional gene? Yeah, well, so I'm reinterpreting this question. So there's two ways of viewing this question. One would be if you're taking a largely complete MAG or isolate genome and doing phylogenetic placement using protein coding genes. That's how our species tree builder works. That's how GTDB works. That's how sort of most of the genome-based taxonomic and phylogenetic classification approaches work. And so those are either going to be clade-specific or universal phylogenetic markers. And they're typically on the order of 50-ish, depending on how you defined it. Universals. And then there's on the order of 100 if it's just bacteria. And then as you go down, it depends on your phylum. So that is the approach taken. And that is, it's tree building, it's multiple sequence alignment and trimming to conserved regions of those multiple alignments and so on. However, if you wanted to use just a single marker gene family or a limited number of them instead of using the full collection of phylogenetic markers, you could certainly do that. There's a tool we have, build a gene tree that you can use on any gene family or a set of homologous genes, but it's not considered an official phylogenetic placement in the same way. Unless you go in with something that's known to not be a horizontally transferred, your gene tree is not necessarily going to align with your species tree, right? So you have to be very careful there. But you could pick any one of the universal phylogenetic markers. Unfortunately, a lot of them are quite short and should probably usually be used together, which is usually going to be possible for a contig because the ribosomal proteins typically cluster on the genome. Anyway, so the version of this question, which is, I have just one contig, it has a phylogenetic marker, can I use that in the absence of all the other phylogenetic markers? Yes, but you have to do it kind of manually. Okay, can we just use assembled sequences for subsequent analysis without a binning step? What would be the pros and cons of each way? And the answer is yes. Annotation tools, binning is a way to get around the limit. Yeah, it's definitely, that's a good point. Just powering ahead with a ton of contigs that haven't been binned, you could do it, but like I said, you really are better off doing binning as a class of fractionation to get the most valuable data out of it. I cannot imagine a scenario where you would want to take high quality mags and take the contigs and just throw them back in the soup. I'm sure someone could come up with something, but it's always going to be better to have it split off into a what you believe is from one lineage. What functional annotation of mags would be considered redundant as opposed to those that are dependencies between retrieved mags? Okay, I'm thinking what that means is, say you have two mags and you have a pathway with known gene families in it, and you're winding up with mag A having half of the gene families from that known pathway and mag B being the other set of system components. Are both of those mags carrying out that pathway by themselves, or are they interdependent species? Or scenario 3, was it a mag that wound up being sort of two medium quality mags that should be merged? Those are three possibilities and deserve a really close look. And, it's going to be dependent on what that analysis looks like. I would say probably if you have separate mags, but the same system components are significantly overlapping, and then other system components are missing, I would say it's probably the result of that lineage not having decent coverage of the gaps in that pathway, the gene families that carry that out. That can often happen. And so I'd say probably the genes are either not in contigs or not recognizable. Another thing you would look for are transporters that carry those metabolites that connect those pathways. So, I would say if it's if you look at the pathway, the sequence of metabolite transformations, and the enzymes are very patchwork, meaning sort of interspersed gaps that are complementary, that's probably a case where you needed to merge the mags. And it's hard to picture that many transporters and that much pathway completeness being handled by a metabolite interdependency between those cells, passing that metabolite back and forth that much through transporters. But if it's the first part is one organism and the second part is another organism, and they both look like high quality complete mags otherwise, then maybe there's a metabolite handoff halfway through. And there I'd look for transporters for that metabolite. Again, transporter annotation, functional annotation is even worse than just about every other class of enzyme or protein. So it's tricky. That's the best I can do for that answer. Are the analysis steps similar to the analysis of barcodes? You can split your sequencing data using barcodes. If you consider, it depends on how you define barcodes. There's so many different ways of using barcodes. I can't answer this. I'm sorry. What's leaping to my mind is lipid emulsions with barcodes and you know small metagenomes and things like that. Sorry, I'm thinking about the wrong thing probably. Which Refseq database to use for phylogenetic placement? Can the user change it? We are, we're updating it. It should be in sync within about a month. That's right. Is there a minimum number of, I should say GTDB is a classification system from the Hugenholtz group, which is available on KBase. And please use that as well. So you can do both. You can do the tree building with Refseq. You can use taxonomic classification with GTDB, which consumes Refseq as well. Taxonomy is another whole can of worms, which we are trying to do some nice reconciliation in KBase. That's a work in progress. Stay tuned. Is there a minimum number of Contigs to get as quality control to confirm a successful Contig binning? Yeah, this answer here is good. Longer Contigs, fewer of them. I mean, it's like isolate assembly. A lot of them, you know, even now come out with, you know, 1,500 Contigs. A lot of times it depends on how many transposons they have that they break on the long repeat, even with decent depth of coverage. So mags, it's even worse. But really, the check-in is meant to be the initial basic quality control of, you know, how complete are your phylogenetic markers based on the lineage assignment. Does MaxBin2 only bin bacterial genomes? Yeah, you can do bacteria and archaea. We don't have a tremendous amount of functionality for eukaryotic microbes. That is something that's being worked on, and more to come on that later. Is there a way of mapping short reads on the mags or assembled contigs? Yes, that's going to be coming soon. It's related to splitting reads as well. Polymorphism, depth of coverage analysis. We're thinking quite a bit about this right now, and we'll hopefully have something for you soon. Annotate multiple microbial assemblies, choose a domain. No, unknown runs both. It's, yeah. Rast, no. Okay, archaea, yes. Protists, no. I really want more reference protists, but it's slow in coming. Bacteria and archaea yeah, a lot more eukaryote and virus questions. We do have some tooling, especially for viruses, coming online, like VerSorter. VerSorter may be released or maybe in beta. I'm not sure right now. Fractionation based on lineage is a whole nother topic that I didn't have time to get into, and we're building out functionality there as well, because it's a great thing to do, obviously. Megahit, metaspades be used for metatranscriptome assemblies. Cufflink string tie. Metatranscriptomes get into expression analysis, which is a tricky business indeed for metagenomes, and we are thinking about that deeply, but it's beyond the scope of this here. Which database for genome classification, assembly, placement, MaxBin use? So MaxBin uses universal phylogenetic markers and scans the contigs for those markers, and it's just based on their HMMs. It's part of the reference data. I think I'm not going to finish these questions in the next 4 minutes. So I don't know how to pick which questions to answer. What causes contamination? Chimeras or poor binning? Both are caused contamination. Heterogeneity and contamination.
There's a few totally unanswered ones at the bottom. Maybe those
were there new ones added since I started at the bottom?
Yeah, maybe. I don't know.
Okay. All right. Benefit to reassembling after binning. Great question. Some people think so, other people don't. So part of that read mapping stuff will serve as a foundation for a tool that's coming soonish, I believe. If I can, anyway, hopefully there will be some automation to take a binned contig, extract the reads that correspond to each bin, and then redo the assembly of that subfraction using an isolate assembler. Hopefully it will be an easy to use. I think I can do it, so it's pretty easy to use, but still powerful. Mixed yeast and bacteria. What can you do with the yeast contigs? I mean, we do have some fungal annotation tooling. So if you can split them off, you can go into fungal analysis pipelines on KBase, but I wouldn't use the bacterial specific stuff. Why would you use Proca? I already answered that. Okay, so it was just the two that were the new ones. All right, well, there's only 2 minutes left, and the rest of these seem like they're filled in, so we'll go over this question and answer thing a bit more, but it'll be available for you to read later. I want to thank the whole KBase team, because I mean, this tooling that we've built is really on top of the foundation, which is the KBase platform, and that's a really tremendous amount of work and really creative thinking and just wonderful stuff. This platform that a lot of people have built. So this is all just applications built on top of it that happen to be specific to particular bioinformatics questions that Sean and I have. But you can really do quite a lot with KBase that we haven't talked about at all, and will be the topic of other webinars. Anyway, between the management and thinking about which directions to head and how to build it, it's really been a massive undertaking, and it's really I'm thrilled with KBase can do, and I'm really thrilled that you guys are interested in using it to further your research. I hope it serves you well. So with that, I'm going to stop sharing. And Ben, do you have any final thoughts, Sean?
I do not. Thanks everyone who stuck to the end. Just like I said, follow us on Twitter. If you have any technical support issues, you can find the link to our help desk on our homepage, and you can create a ticket, and we will follow up with you there. Thanks everybody. Thanks Dylan and Sean, and we'll talk to you in another month for next webinar.
Thank you, everyone.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE_KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.